Hi everybody. How you doing? Crazy times. Today we are having a crazy day. Schoology's down, castle's down. I'm still standing. So, YouTube decided it didn't want me to go live. I'm going to have to figure that one out. So, I'm going to do some review on here. So, AP Bio, if you're ready to do some review, we are on Unit 2. So, I am going to do this. Tell your friends that I'm on here. It was hard to let everybody know, like, hey, I'm doing this live. So, tell other people if they're in AP Bio, go watch MERM right now. I will save it. It'll be on my story. I will record it as long as my phone has enough space. I'm a little bit worried about that, but I will try and save it and put it to my YouTube. So, I'm going to go old school like my classroom with the big boards, and I'm going to go right through Unit 2. So, if you go to my Schoology and you go into this week, the second week of this, March 30th. No, we're week 3 of this, March 30th. There's a Unit 2 big idea sheet in there. So you're going to want to pull up that big idea sheet. If you can print it, print it. If you can't, just maybe grab a notebook, do it in your notebook. So the first thing is to draw and label an animal cell, write in the functions, and show the interactions. So you guys know my really big boards. I'm going to make the entire board be my animal cell, and then I'm going to zoom into different areas. So first of all, I'm just going to draw a nice big cell. All right, there it is, one big cell. And now I'm just going to go into the middle and I'm just going to do a nucleus. So this right away tells me that I'm talking about a eukaryotic cell because it has a nuclear membrane. And this came to be because a prokaryotic cell took in another cell, and that became an organelle that we now call the nucleus. It has a double membrane. So you'll see I drew 2 parts around it. So this double membrane, it has 2 membranes because this was the original cell's membrane and this is the cell that took it in, its cell membrane. So when it engulfed it and took it in, did endosymbiosis, that's what happened. So the nuclear membrane does have pores. Let me use some different colors here. The nuclear membrane does have pores, which are little openings. And they're pretty small, so they will let RNA out, but DNA can't get out. So here's my DNA. It looks all stringy. We call it chromatin. So when it is not condensed and it's not clearly DNA in separate chromosomes that you can tell one from another, you can just call it chromatin. And there are regions that are pretty dark and dense, and that is the nucleolus. So there's my nucleolus right there. So I have my nucleus. It has a double membrane around it because it used to be its own cell. I've got my DNA. I've got a nucleolus. The nucleolus is where RNA is made. So that means this is where transcription occurs. And it's also where proteins come together with our RNA to make ribosomes. All right, so all of that is going on in my nucleolus. So I want you to just think about the process of transcription. We're going to take a little bit of this DNA, wherever there's a transcription factor at the promoter, at the Tata box, and we're going to make just one short section of RNA. So that blue is representing my short section of RNA. Then it gets modified before it leaves. So it gets a cap, it gets a tail, and then any of the introns will be cut out of it. And then this piece of RNA, messenger RNA, is ready to leave. So it can fit through that nuclear pore and leave the nucleus. So that's about all the things that we really had to talk about for the nucleus. Now we can go outside of the nucleus. So that mRNA is going to go to a ribosome. So here is a ribosome. They are extremely small and all cells have them. So it doesn't matter if you are a prokaryotic cell or a eukaryotic cell, you will have ribosomes. Some of them are free, which means they are not attached and they're just all throughout the cytoplasm. Usually anything that is a free ribosome, that is used to make proteins that are going to be used in the cell. So make proteins used in the cell. If I have a free ribosome, it's making a protein that's going to stay right here in this area. But let me grab another color. We have ER, endoplasmic reticulum. So here is my ER, and it goes back and forth like a little system of tunnels. And there tends to be a lot of ribosomes on certain sections of it, and that's called rough ER. So rough ER has the ribosomes, and these ribosomes are going to take that little piece of mRNA. They're going to go through the process of translation, and they are going to make a protein. So this black right here, this is representing a protein that was made by the ribosome on the rough ER. And the rough ER is going to transport that protein and really keep it protected. So it protects it from any enzymes that are out here in the cytoplasm. And this protein can now move through in its protected sense and make it into a vesicle. So at the end of the ER, I'm going to have a vesicle here, and I'm going to package up my protein inside that vesicle. So again, it is protected. The next place it'll go to looks like a stack of pancakes, and it even looks like little syrup dripping off the ends of the pancake. This is a Golgi body. Oh, and this I would label a vesicle. So a vesicle is just a membrane sac, usually coming off of ER or off of a Golgi body. And the Golgi body is now going to take in that protein, but I'm going to change it a little bit. I'm going to change the shape of it, and I'm going to change how it folds. So the Golgi bodies modify proteins. Let me give you an example. There are glycoproteins in the plasma membrane, and glycoproteins have both a protein component and a sugar component. So the Golgi bodies would add that sugar component onto it. So to modify it means they're changing that protein or allowing it to fold in a certain way. Eventually, this special protein is going to make it into another vesicle, and it's going to go through exocytosis, and it's going to leave, and now it's going to be outside of the cell. So that entire thing that we just did, that's called the endomembrane system. So the endomembrane system allows a protein to be made, packaged, protected from cytoplasm and other enzymes, and modified, and then eventually exported from the cell. So if you have a ribosome that is on rough ER, it's probably making a protein to be exported by the cell, not to be used within the cell itself, but to go outside of the cell. All right, so far we've drawn our nucleus and our DNA and a nucleolus. We've made messenger RNA. That's what was up here, messenger RNA. We had the messenger RNA leave through a nuclear pore and go to a ribosome and make a protein through the process of translation with the help of tRNA. We talked about how this ER is rough because it has ribosomes on it. We talked about vesicles, we talked about Golgi bodies, and we talked about exocytosis. So now I'm going to move on to another part of my cell. And let's say that I have something outside of the cell like starch. Starch is too large to go through the lipid bilayer. So the lipid bilayer would actually come around and engulf it and take it in. And so once it's in, it's got the membrane of the cell around it, and it's got the starch inside it. It would fuse with an organelle that you usually find very close to the edge, and it has a lot of enzymes inside of it. And so this is a lysosome. So these two will fuse together, and the enzymes, we call them hydrolytic enzymes. That's because they do hydrolysis. Hydro, they add water. Lytic, they break apart. So these are hydrolytic enzymes. They will digest the starch, break the starch all up into glucose monomers or sucrose or fructose or whatever smaller sugars it's going to make out of that starch. All right, lysosomes you usually only find in animals because animals are taking in foods. Plants make their own food, so they make G3Ps. So we really don't need these in a plant cell. Okay, sorry, I'm reading in the comments. What does it say under nucleolus? All right, it says RNA is what I would find in the nucleolus. Transcription is the process of making RNA from DNA. And then proteins combine with our RNA and actually build ribosomes. So that's what it says under there. Let me just see if there's any more comments. You guys can definitely ask me questions as we go. The whole idea, the whole suggestion for me to do this live was for you guys to have a chance to ask me questions as I do it. So definitely let me know if you have questions. I'm so glad that there's lots of you on here too. Hope you guys are having a good day. All right, I am going to keep moving. Let's see. I'm going to make a mitochondria. And so one of the things that I want to tell you is that on the AP exam, I want you to avoid phrases like powerhouse of the cell. People that grade AP exams really hate that kind of language because it doesn't really show that you understand. It just shows that you know a very typical American meme for mitochondria. So mitochondria have a double membrane on the outside. That's the first thing you should know. For the same reason that a nucleus did. This used to be its own cell, so this was its own cell membrane, and this is the membrane of the cell that took it in. But it also has a membrane on the inside, and the membrane on the inside is called the cristae, and it really increases the surface area for electron transport chains. Electron transport chains are those specific cytochromes, those proteins in the membrane that are going to be able to take NADH and FADH2 and get their electrons and carry it through. So when they carry it through, they're able to build up a gradient. So if I make this look like, so you can see the inner compartment, it'll build up H pluses. And the H pluses build up in the outer compartment. That process is called chemiosmosis. And it's all due to the fact that the electrons are moving through the electron transport chain. And when electrons move through the electron transport chain, the last place they end up is with oxygen. So if you take an oxygen and you take 2 electrons and you take 2 H pluses, you make water. So the process of respiration does make water. Got to just lift this a little higher. All right, it does make water. But the most important thing that it makes is ATP. So the process of making ATP is going to utilize this H plus gradient. So I need an ATP synthase. That is my special channel protein that has the spinning end on the inside, the rotor on it. It'll allow the H pluses to move down their gradient from high to low, outside compartment to inside compartment. And as it moves, this part turns and it puts together ATP. So the real importance of the mitochondria is to allow that electron gradient to build up all those H pluses. So the moving of the electrons is really, really important to allow me to make a lot of ATPs. All right, I'm going to keep going around in my diagram, and now I'm going to do, these are a special set of microtubules. They are called centrioles. There's 2 of them. They usually look like rigatoni, and they go perpendicular to each other. And these are what's going to double, go to the ends or the poles of the cell in order to form spindle fibers, so that during anaphase, you can separate chromosomes. So that's the purpose of a centriole, to be able to pull those apart. There are other microtubules. So this is a microtubule organizing center, which means it's got a lot of microtubules doing a very specific job. But there also can be smaller microtubules like this along the edges that sort of just give it support and shape. So these are microtubules, or if they're even smaller, they can be called microfilaments. So a filament is as short as you can get, or as thin as you can get, and a microtubule has multiple microfilaments together and is a larger structure. All right, let's see if I'm missing anything. Oh, outside of the plasma membrane, so I'm going to eventually do the plasma membrane, just not in this first diagram, but I'm going to add some little hairs along it. So these little hairs would be cilia, or if I had a really, really long one, that would be a flagella. And those are for movement. Those allow the cell to like swim, to move to another place. So let me just check this over. Oh, I know of something else I could add. I could add a vacuole. So a vacuole doesn't have to be next to the edge. Usually the way you can tell a lysosome versus a vacuole, lysosome's usually close to the edge so it can join with whatever comes into the cell. But if this is my vacuole, this is for storage. So based on what type of cell I am, if I'm making an abundance of something, I would use this vacuole to store things that I have extra of. All right. I think that's a pretty good thing. I think we can move on. If you think of something else that you're like, hey, Merm, you should have included that in your cell, tell me and I can go back to it. But for now, I'm going to grab another board and I'm going to go on to the next page, which is doing a plant cell. So I'm going to start all over again, but this time I'm going to make it a square. And the outside, I'm going to make this all fibers. This marker is awful. So these fibers along the outside, that's my cell wall. And this is made of cellulose. Don't know if you can even read that. This marker I gotta chuck. All right. Cell wall. It's made of cellulose. All right, so it's got a lot of fibers here on the outside. Tends to be pretty rectangular in shape. All right, inside of that, I want to have my cell membrane. So here's my cell membrane. So that's my phospholipid bilayer. And then the next thing I want to show is a vacuole, because plant cells tend to have a large central vacuole. Now, the water that's in here pushes out to every side. That pushing is called turgor pressure. So plant cells love to be in a very hypotonic environment, because hypotonic is going to constantly make water move in. And if water moves in, you increase the amount of turgor pressure. More turgor pressure from the vacuole, the more it's going to push on the cell wall, or the cell membrane, which will push on the cell wall, which will hold it up and give it shape. So turgor pressure is really important. Water is really important for a plant, for its structure. All right, I'm not going to go through all the parts if I've already done it before, but this could be my nucleus, this could be my DNA, this could be my ER, these could be ribosomes, this could be a Golgi body, and vesicles, and this process of taking a protein made at the ribosome, passing it through, modifying it, and exporting it out of a cell. That would all still happen here. Definitely still mitochondria. So there's a mitochondria making ATP, something you find in all cells. Cytoplasm, all of this out here is my cytosol or my cytoplasm. Definitely important for a lot of reactions, especially things that all cells do. So for example, glycolysis, every cell does that, so that happens out in the cytosol. All right, but now I gotta move on to the organelle that's super important for plants. So if this was a cell that was in like the Palisade mesophyll, I could have about 200 of these. So this is a chloroplast. It also used to be its own cell, which means it's got a double membrane. The inner membrane was from its original cell. The outer membrane is from the cell that took it in. And a chloroplast have stacks of grana inside of them. So this is my grana. This is where the light reaction happens. So I will eventually be going through photosynthesis, but if I just go through it quickly, I've got light coming in. Light is exciting electrons within photosystem 2. Those electrons move through an electron transport chain and end up producing NADPH. That movement of electrons also allows me to build up a lot of H pluses inside that thylakoid space. That's going to be really important because I can let those H pluses flow out through an ATP synthase and I can make ATP. So the production of NADPH and ATP are crucial in the light reaction. Hydrolysis also happens where H2O is split and oxygen gas is formed. So those three things are the three products of my light reaction. But then I also have the stroma, which is the liquid part of the chloroplast. And in the stroma is where the Kelvin cycle happens. So photosynthesis, 2 steps. Light reaction is step 1. Kelvin cycle is step 2. So in the stroma is where carbon dioxide comes in and it gets fixed. And eventually this process is going to make G3P. G3P are 3 carbon sugars that you can put together to make glucose or fructose or sucrose or even eventually something like starch. So the Kelvin cycle has 3 steps: fixation, reduction, regeneration, and produces G3Ps. All right, so now we have our plant cell diagram done. So I got to go back over here to my computer and see what comes next. All right, sorry, I had to log back in. All right, important cell concepts. So let me just grab my computer here so I can be reading these off to you guys at the same time. All right, what are the key differences between prokaryotic cells and eukaryotic cells? So let's take another board and let's do prokaryotic cells and eukaryotic cells. So prokaryotic cells definitely came first. These have cytoplasm. They have a membrane around the outside, a cell membrane, a plasma membrane. They have ribosomes. They can have microfilaments or microtubules. They can have cilia. But that's about it. They're pretty simplistic in their structure. And they, because of that, tend to be very small in size. Very limited because of the fact that they don't have any organelles that have membranes around them. Eukaryotic cells were a prokaryotic cell plus it took in another cell. So this is called endosymbiosis. Endosymbiosis, to be symbiotic means to live with another organism. So endosymbiosis describes the process of how eukaryotic cells came to be, prokaryotic cells taking in other cells. There is proof of this, and the proof is because they have 2 membranes. One is their original, one is from the cell that took them in. From the cell that took them in, you get the second membrane. They also have their own DNA and their own ribosomes. And because of this, we have compartmentalization. All right, compartmentalization is a really important word because compartmentalization means that you can have reactions going on here and here, and they don't mix. So if they're not mixing, you can really separate the enzymes and the processes, and you have a really great chance that you're going to be far more efficient, which means cells can now be much larger. One thing that is really important about a lot of the organelles and a lot of the cell is that they have increased surface area. So the most common way you do that is by having folds. So even if the overall cell ends up looking something like this, it'll have a really high surface area and a really low volume. That allows for the maximum amount of diffusion and osmosis to occur for your cell. And you still can have all these different organelles, some of them all different shapes, but you can have all these different spots within your cells that are doing different functions. All right, so I think I hit a couple questions with that. So the first one was key differences between prokaryotic and eukaryotic cells and endosymbiosis. Compartmentalization is important because it allows reactions to be separated and enzymes not to mix, makes it far more efficient for a cell. All right, next one, components of the endomembrane system. So I actually did that in the first drawing, so I'm going to go back to the first drawing. And again, if you guys are having questions, please just let me know. I'd be happy to answer your questions. I will put this on my YouTube so that you guys can go back and review. It's just going to be really important over the next few weeks before the AP exam that you guys really take the time to go back through all these units and make sure that you remember all of this information. All right, so let me grab my first diagram. I'm glad I didn't erase it yet. I almost erased it. Okay, so the endomembrane system means how do you keep the protein that you made enclosed. And so here at the ribosome is where proteins are made. So that was my messenger RNA coming in, and the black was the protein that was made. And so it goes through the rough ER and it stays protected. Then it goes into a vesicle and then it fuses with the Golgi body. So now my protein is within the Golgi body. That also modifies it as well as protects it, puts it in another vesicle, and eventually lets it outside of the cell. So that is the endomembrane system. Okay, surface area and volume. This one I'm actually going to skip for right now because I don't have a calculator downstairs here with me. Here I am thinking I'm so ready for this Instagram Live and I can do AP Bio. I do not have a calculator right here. So if you haven't, I'm going to do this. If you have any troubles with calculating surface area, volume, or surface area to volume ratios, let's just do it on the Zoom on Thursday. So every Thursday at 1, we'll Zoom and we'll go over that unit. So this is unit 2. Let's go on to the plasma membrane. All right, more drawing, more things. So I have another board. Gosh, they let me go in that first day. You know that first day, you guys, that first crazy Monday, and they said, hey, if you come into school between 8 and 3, Mr. McCabe will go get all your things from your locker or classroom or whatever. Thank goodness for that. I grabbed about 7 of these boards. You know how much I love these boards. So if this is my cell membrane, what it's really made up of are phospholipids. So a phospholipid has this polar head, and polar means it's going to be hydrophilic, and it has nonpolar tails, which means they are going to be hydrophobic. So out here, there's water. So if I'm outside of the cell, I have a water environment around me, this polar head has a negative charge to it, which means it's going to be attracted to water and fine in water. But the tails don't like water, so they form a bilayer. So I would have to keep drawing this the whole way around. The heads are always facing out, the tails are always facing in, like that. Each time I draw a kink in the tail, I'm showing an unsaturated fat. So because there's unsaturated fatty acids in the tails, it allows it to have a kink. So unsaturated fatty acid tails have kinks or bends which allows it to be more flexible, which is going to help when you want things to be able to cross through. The other thing that you find in the lipid bilayer, only by the tails, is cholesterol. So cholesterol is 4 fused carbon rings. So here's my cholesterol. Cholesterol wedges its way in between there and also increases the flexibility of the membrane. Oh, I might as well use that word. If you have a region or a molecule that has 2 regions, a hydrophilic region and a hydrophobic region due to polarity and non-polarity, it's called amphipathic. So the fact that a phospholipid, which I really didn't even write that word on here, the fact that a phospholipid is amphipathic is why it makes such a great membrane. All right, but there can't be just this because there's not a lot that can get through just this. So let's talk about what can get through just this. If I was oxygen gas, OO with a double bond, that is definitely nonpolar and it's definitely small. So the tails like me, so they let me through. Carbon dioxide, same thing. They like me, so they let me through. As long as I'm going from a high concentration to a low concentration. So that is passive transport because it doesn't use any energy, and it's simple diffusion because it only requires the molecules to slip through that phospholipid bilayer. Now I'm going to show you facilitated diffusion. So I'm going to make in my lipid bilayer a big protein. So here's my big protein. And my big protein is actually a channel. And this channel is very specific. It will only allow certain molecules through. And so we'll say that this channel has a gate on it. So right now it's closed. I'm just going to remove this word to give myself a little bit more room. But we'll say that this is a sodium gated channel. When the cell receives a signal, the gate opens, and now sodium, even though it has that charge on it, can now pass through. It's got to move from high to low. Facilitated diffusion is still passive transport, which means it doesn't use any energy, but it is moving with the gradient into the cell. And this protein is called a channel protein. And channel proteins are also amphipathic because they actually have regions. This region right here has to be nonpolar because right next to it are a bunch of tails. So it has to like it. And then this region is going to be hydrophilic because it likes the heads. So it's weird to think about it, but those proteins actually are amphipathic as well based on where their location is. There's got to be some parts of it that like water and some parts of it that don't. All right, the next thing I want to draw is a pump. A pump is not facilitated diffusion and it's not passive transport. A pump usually has a very specific shape and it can actually change shape. So a great example is the sodium-potassium pump. And it is a user of energy. It uses energy to do active transport, and it will allow you to bring molecules against their gradient. So I just had sodium come in from high to low. If I wanted to move Na plus back out, it's going from low to high, and so that would require a pump for it to move. And some pumps can move 2 molecules at the same time. So K could move this direction from low to high. And so that's how the sodium-potassium pump works. All right, it will bind ATP to it, break the ATP into ADP and PI, and it will use the energy from breaking that bond in order to pump the molecules against the gradient. All right. Later on, we will have receptors and recognition things. I just want to do one more thing today for you. The last thing on the bottom of this page is how will each of the following molecules enter the cell? All right, so oxygen. Oxygen is definitely nonpolar and small. So this is going to enter the cell through the phospholipid bilayer. So that is passive transport, and we just call that simple diffusion. All right, the next one on my list is a sodium ion, Na+. And I know that outside the cell, that's in high concentration. It has a charge, so it cannot go through the phospholipid bilayer. It has to go through a sodium channel protein. This is called facilitated diffusion. And facilitated diffusion is passive transport. It does not use energy. All right, the next one, water, H2O. Now, water is polar, but water can enter two different ways. So I'm going to have two different ways here. The first one is by simple diffusion. So going through passive transport in the phospholipid bilayer. It can actually bend a little bit and slip through, but it's not a great way to get a lot of water through. The best way to move a lot of water at once is called an aquaporin. And an aquaporin is a channel that is specific for water. Someplace that you would obviously see a lot of aquaporin is in your kidneys, because in your kidneys, you've got to move a lot of water in a short amount of time. All right, next one on my list is testosterone. So first I got to think about what testosterone is. Testosterone is a steroid. It's a sex hormone. And so because it is a steroid, it is nonpolar. So testosterone, steroid. This will be able to go right through the phospholipid bilayer. That's passive transport, that's simple diffusion. All right, so because testosterone's nonpolar, it can get right past those tails. All right, an amino acid. Amino acids are polar, which means they usually need some sort of channel to get in as long as they're going from high to low. They could use a pump if they're going low to high. There are even some like co-transport models that show that you can move one thing in with another. So even though it might be hard to bring an amino acid in, you can with the help of the transport of something else. Maybe one is moving out and the other is moving in. All right, a potassium ion. So that's a K+. Now the K+ is low outside of the cell. So if I want to move it in, I am going against the gradient. So to go against the gradient, I need a pump. So to move potassium into a cell would be to do active transport using a pump like the sodium potassium pump. Active transport. All right, my next one is starch. Starch is really big. Starch is a polymer, so you can't go through the bilayer, you can't go through a channel, you can't go through a pump. This has to be brought in by endocytosis. So endocytosis is like this. If there's the little molecule, my cell has to go like this and engulf it and surround it and take it in. That's the only way you're going to get something large like starch in. The next one, epinephrine. Epinephrine does not enter, so I'm just going to put a big X. Epinephrine is a ligand, and it is received by the receptor, and then the receptor will change shape and work accordingly, but the epinephrine doesn't actually end up entering the cell. All right, and then the next one I put was a plasmid. And remember, prokaryotic cells do the act of transformation, which means they will take in things from their environment, and they do that by endosymbiosis, so a lot like endocytosis. So a lot like the picture that I drew with the starch. And so a bacterial cell will take in other things from its environment, especially plasmids from other bacteria. All right, so I'm just going to scroll forward a little bit. The next things to come are all going to require me to have my calculator. So I'm going to do another round of this tomorrow. I will post this one that I did right now. But tomorrow I'll go through osmosis, I'll go through facilitated diffusion and active transport and all of these things again. And then I'll go into water potential. And I'll do a bunch of examples with you and sample problems with you of how to solve water potential. And then when I'm done with that, I will go through the labs that we did. So we did some AP biology labs on how to calculate water potential for potatoes or sweet potatoes or carrots or whatever you have. So we'll go through those labs. We'll go through the diffusion osmosis labs with our little cells that we made too. All right, thanks for joining me. Have a great day, everybody.